Hi everybody, this is Dr. Ed Park from Recharge Biomedical, and today we're going to be talking about comparing exosomes. On the left we see René Magritte, surrealist painter, and he's famous for making this painting that says "Ce n'est pas un pipe," which means this is not a pipe. Of course, we don't really know what he was getting at, but it's a picture of a pipe or painting, and it's important to realize that facsimiles are not the thing in and of itself. Very important disclaimer. I have no way of knowing if the validity of the data that's going to be shown is real. Again, having said that, I have no reason to doubt it. I am not a molecular biologist. I may make some misstatements. This is not an advertisement for Kimera exosomes, but it's an education for people. I get asked a lot, why don't you just use so-and-so brand? And hopefully this will explain to them. As far as conflicts of interest, I do sell Kimera products in my practice, and I do make some money off of them. I did not receive, nor was I promised, any reimbursement for this webinar. So we talk about wine. There's the really good stuff, Screaming Eagle, Chateau Lafite. Then you got the stuff you can buy in the liquor store for $3-4, Night Train, Thunderbolt. Are they the same? Maybe not. You could even buy, as long as they don't have added benzene or these things that they added during Prohibition, you could buy ethanol, 200 proof, and add grape juice and talk yourself into saying you're having wine. When I first embarked on this data set that Marielle Gonzalez from Kimera presents, I thought it was going to be a comparison of sumo wrestlers, little sumo, big sumo. But in fact, we may have something quite different, even actually worse. They might not compare at all. So the goals of the talk will be to describe what are exosomes, what are they made of, how are they made, how do you know you have them, and can we compare the potency? All right. Real briefly, what's a mesenchymal stem cell? Well, a stem cell is what you became when your sperm and egg got together. You were one egg, and now you're 7 trillion cells. In certain areas, some cells are disposable. Others are relatively immortal by telomerase action. That's shown in the bottom left. And a stem cell has the ability to replicate asymmetrically, meaning they make a more differentiated daughter and then a regular stem mom, and they keep on doing that, thereby allowing themselves to renew and not die or differentiate. Slide we show a lot. I've adapted. What are exosomes? On the upper left, you see exosomes going out. There are a lot of pathways you don't really understand, of course, starting with the protein and manufacturing and the Golgi apparatus, which is adjacent to the endoplasmic reticulum. Anyway, they're packaging these little packages into bundles, multivesicular endosomes, and they get released in these bunches, like little bunches of grapes. So they go out. In the middle, we see they come in too. Nobel Prize was awarded for these clathrin-coated pits. So part of the role of these markers to my left, CD63 and 81 in particular, are more specific to exosomes, is to interact with surface proteins and sometimes particularly even the clathrin-coated pits. So exosomes go out on the upper left and they come in, and this is the communication, default communication of the cell biology. So of course there are 100 nanometers around there, about 1/1000th almost of the diameter of the actual cells. They're not cells, they do not reproduce. They don't have their own machinery for energy, for DNA, etc. They don't have their own endosomic pathways. They're just little messages. And although the slide above me shows MHC1 and 2, this appears to be more from cells that are derived from the immune system. Exosomes from the immune system. Your run-of-the-mill exosomes, at least from the MSC type, which we've studied the most, do not have. So on the upper left, we see exosomes should be around 100 nanometers diameter. They have the tetraspanins. In the middle, when we go by size criteria, because anything that is a phospholipid bilayer outside the cell is like a vesicle, an ECV, extracellular vesicle. So there are probably things in the 2 to 1,000 nanometer range that are doing something else besides this exosomal signaling, this typical one that we want to get at. And then, God forbid, on the right, apoptotic bodies, when cells are destroyed by themselves or other mechanisms, they become these giant blebs. So you can get things on the order of several microns. They're just thinking about like the dead debris, the flotsam and jetsam from a destroyed warship. Excuse me. So we have a situation where, like Dr. Frankenstein said, it's alive. We have to always respect and understand that cells are alive. They have to be taken care of. I was reminded that animal husbandry, it means husband is to take care of. Arguably, if you're going to do this, there's a controversy. And I've had people say to me, well, why do you keep on using the same placenta? Aren't you going to run out of placenta? And that belies a good question that is founded in a misunderstanding of cellular biology. And we'll get a little bit into that. But what we see on the upper right is a standard slide from Thermo Fisher. This is someone taking a cell line out of liquid nitrogen and then warming it in a bath. I do not know what Kimera does. It's not my business, and I presume or I infer that they are basically taking these cells from a master mother line that has been frozen in liquid nitrogen, and every so often, as often as they need to, they take it out. They thaw it as above. And it's like a Star Wars clone, Boba Fett. They have a batch. And so when they thaw out a batch, it's day zero. And that will go to the question that people naively ask, well, aren't you going to run out of cells or aren't they going to go bad? It's quite the opposite. The people that are running the same cell line in perpetuity are getting bad stuff. And that, spoiler alert, may be why we get different results. On the bottom left, if you want to continue the wine analogy, there are certain things that affect wine or grapes. Of course, they're called terroir in French. So the climate, bad season for the grapes could be a good season for wine. But in the process of cell biology or cell husbandry, we have things like, oxygen, CO2, I guess even light and sound, arguably. So you can whisper to these cells and make them differentiate into different types. That people are very proud of that. But the way that you care for them, how they're growing, and what kind of, let's say, three-dimensional matrices, all very, very important, and all would be unknown unknowns if you decide to go into making exosomes. So another analogy would be chicken farming. The eggs come from chickens. The cells are alive chickens. Of course, if you start to poison all the chickens, or the chickens are no good or old, you're going to get bad eggs. Everybody with me, I hope. So more of the nitty-gritty. Let's say you say, wow, what's so hard about making exosomes? I'm going to do it myself. Well, here's just a short list from Thermo Fisher about the stuff you're going to need. On the left, you're going to need a cell culture hood, incubator, water bath, centrifuge, refrigerator, freezer, cell counter, inverted microscope, liquid nitrogen freezer, sterilizer, etc. You're going to need more stuff, more cytometers, microscopes, pH meters. You're going to need people who are trained and expert in doing this. These are just some typical cell culture flasks. And here's a person readying them for centrifugation. Above me is the core concept you need to understand. As you let cells reproduce, they're stem-like, they can double and double and double. And because of geometric doubling, they call it population doubling, PD. You get a log phase where there's exponential growth. So to answer the naive question, well, how can you use the same placenta from years ago? Well, because you can expand them very rapidly in cell culture as long as there's not contact inhibition, as long as they have room to grow. And so you can expand. And if indeed some people have been more adventurous, they've gone to different countries and gotten their own cells expanded, arguably not good cells, they're senescent. And so you can get millions and millions and get them fed back to you. In this country, the FDA has deemed that to be above the level of minimum manipulation and is therefore not legal. Interesting feature of the curve above is this sort of down curve, and there's a plateau and there's a decline, and we'll get a little bit into what the decline means. Again, into an area which is not my purview. I have not been told, nor do I know, but if you look it up in books and articles, these are general ways that you can isolate exosomes. So as Duncan Ross says, he used to throw away the stuff, the medium and all the stuff the cells were creating. Now that's the good stuff. That's actually the wine that we want. So one typical way they do is they do ultracentrifugation, which is what? Upper left, we see that if you spin something in one of those little plastic aliquots at 300 Gs, at 1,000 Gs, 100,000 Gs they can get from these things, you get a separation based on density. And so you want to get that pellet at the bottom, which has your pure exosome by small density. Makes sense, right? So the bigger particles are on top and the small particles on the bottom. Another commercially available technique is prefabricated ciliated filter cylinders. So what's shown here is like they look like high rises with little afro combs sticking out of them. Well, actually, as you filter through, the big cells obviously won't go into this. Debris won't go into this. So apoptotic buys macrovesicles, like in the 3 to 800 nanometer range. Proteins will, of course, be okay. But inside this 2 micrometer or 2000 nanometer cylinder, you have these little fine hairs. And so in the 200 nanometer range is right in that wheelhouse where it's the right diameter to trap what you want, exosomes. I don't know if Kimera uses that. They don't tell me. I don't ask. Um, Below to the left, we see something a little different. This is, I know, one of the manufacturers uses this. It's called bead agglutination. So what we're seeing here is they use a bead with some kind of protein to, like, almost like antibodies of the natural protein. They use it to trap and identify. And the problem with this is, of course, you can get the residue of the beads and the trapping antibodies or proteins, which can lead to unpredictable results. Not recommended, and as far as I know, not used by Kimera. Slide from last week's presentation, the song doesn't remain the same, meaning cells have autonomy, they're alive, they have the ability to play multiple songs, and these 3 articles are just a few of many, many hundreds of articles looking at clinical conditions. And so when you analyze what's in the exosomes in different disease states, it can vary a lot. And the example I like to use is malignant transformation. If you get somebody who has breast cancer and you give their exosomes to a woman without breast cancer, you can start to show signs of malignant transformation. Conversely, if somebody has liver cirrhosis or kidney failure and they're getting better, something saltatory, something good in those exosomes can help people with that chronic condition. 2 articles, blogs I wrote. The first one was in response to a hit piece by one of the competitors that said, well, the Kimera product is just normal saline. Well, of course it is, because it's embedded in normal saline. And unless you do the step to melt the phosphate bilayers and open up those, let's say, supermarket toys, you don't know what's inside. It absolutely should come back as normal saline. Shouldn't come back as bovine calf serum or any DMSO or anything like that. Another blog was 22 when we talked about all the bad press. People, when there's money at stake, there's long knives out, and people do disparage other people's brands. And I've been asked by other doctors, well, why don't you use this brand? It's great. I'm having good results. Well, good for you. If you're having good results, that's great. I'm going to use what works for my patients. Here's a graph of expression of mRNA. We won't belabor it. You just see that it's not random, the distribution. This musician of the cell knows what it's doing. It's singing certain notes over and over again. We just don't understand the music theory. There's insulin-like binding proteins, BMP. I just read an article about regrowing teeth with BMP. VEGF is an endothelial growth factor, very important. So a lot of interesting, cool stuff going on there. Tissue inhibition of metalloproteinase, another important thing needed for healing of musculoskeletal tissues. About 3 years ago, there's an article by Ferguson and Nguyen, and she has a great YouTube video explaining this. And they looked at the microRNA, which may be the hidden stars of exosomes. It's not so much the proteins or the messages, but actually it's the microRNA, which are these short sequence hairpin sort of blockers. They specifically block certain messages. So in the case of neural dedifferentiation, there's one that we talked about in the nerve regeneration webinar. They can allow for expression briefly of a microRNA that will block the dedifferentiation inhibitor. What does that mean in English? It means the thing that's keeping you from going, S or what ludicrous mode in your Tesla is gone. And so what happens is the cell's able to act young and dedifferentiate, which is really exciting. Anyway, if you look at the complexity of this, you look at the 3 Venn diagrams, the upper left, the MSCs, human embryonic kidney, and I think it's a prostate cancer. You see that it's not random. The microRNA are different. There's some overlap, but there is specificity, which can then be by machine learning abstracted into biological pathways and biological processes, way above any of our pay grades. So we see some of the readouts from Kimera's two products, the exosome product, the amnio product. We see again BMP, VEGF, some of the same and usual suspects. Insulin-like growth factors, neural growth factors, stem cell factors, all the good stuff you'd imagine would be in there. So just really a kind of simplified version of what we're going to talk about in this last third of the presentation. If you have a bunch of beads, like I don't know if these are styrofoam or candy, but you analyze them under a microscope, all you know from the standard tech of all these companies is how many particles you have and how big are they, and it comes out as like this graph. Yeah. So for example, 100 nanometers is that big peak there, and then under 200 there's another little peak, and it's quantifying by concentration. It's basically a scanner. So you have a microscope, you put the medium on that platform, and the computer does the counting. It gives you a scatter plot and one of these graphs. Yeah. So we get this. It's pretty simple, and everyone reports their particles. And herein lies the rub. People say, well, doc, you can get same thing for 30% less, and you got 5 more billion. And that is really the crux of what we're trying to answer. Do you really have 5 more billion? The technique that Marielle used here was CD81. We know that 81 and 63, these differentiation markers, are expressed in canonical or regular ordinary exosomes. So what a Western blot is basically they denature the proteins, they separate them out so they don't have their tertiary and quaternary structure, and then they run them in a gel. Each sample runs in its own column in this gel, and they have some usually hydrostatic force that draws them. So based on the existence of polarity, they'll run it like different horses or dogs down a track, and we get an antibody that's specific for CD81. Afterwards, we light it up, put it on a piece of paper and bind it and light it up, and you see where the CD81 lies. Similar thing, I think this is a RNA gel agarose thing on the left. I believe it was prepared with this RNA kit from Thermo Fisher, but I don't know for sure. There are kits that have the detergent needed to crack open the toy ball so you can release what's inside. What we see is just a screenshot phone to phone from Marielle to Duncan, and it just shows that their 3 products in lane 1, 2, and 3 have tons of this RNA, whereas the redacted competitors 4 through 9, not so much. And the control is in lane 9, again, lighting up. HPLC, high pressure now, high performance liquid chromatography, just means that you put something in a skinny little tube and you force it through like 50 to 300 atmospheres of pressure. And the speed with which it goes through or elutes through these beads, they're little micro beads, is determined by their molecular size and other things. Anyway, when they come out the other end, the red comes out, or the blue comes out first, then the red. Anyway, they separate differentially as they're being forced through this tube. That's liquid chromatography. And so, for example, if you do a drug test, you see above me this person has cocaine, bupivacaine, Norcaine. They all come out at different times, 613, 635. They come out of the tube differently based on their molecular structure. This is a really popular forensic technique. It's one that I use whenever I make my recharge. We always get an HPLC report to show that the purity is there, the concentration, and etc. Particularly, there are various forms of HPLC. The one that they refer to, and which I'll talk to Marielle about, is called size exclusion, meaning there's something in here that will preferentially disallow certain sizes, and that's how you get these different peaks. So it's HPLC with a twist. So they're actually excluding certain sizes. As we wrap up my part of the presentation here, remember that another of these things, primum non nocere, as a doctor, we don't want to hurt people. We don't want to rip them off, but we don't want to hurt them. And hurting them could include infectious disease. Every report I get has much more than this readouts of infectiousness from this one donor placenta. And of course, it would be crazy to think that that one donor placenta in the freezer, no longer even there, just the cells somehow acquire. But nevertheless, they go through the motions and they test for these infections. If it wasn't there originally, it's not going to get there. There is no placenta. There is no chance to get infected. But I've heard this crazy argument from other manufacturers. People say, well, it's great to have a variety of donors. Well, is it? If we look at this chart, people who have sexual partners, if you have one sexual partner, then you're only exposed to one person, assuming they're the same. Two people who've had two sexual partners, you're exposed to three. But it starts to get a little sketchy when you have four partners, your partners have four partners, you're sharing the infectious risk from 31, etc., and so on, and they tell two friends, and it's not a good story. So when you talk about points of failure, infectious risk, I still have yet to ascertain or understand the argument that having more donors is better. So Duncan is on the call, as is this lovely lady, Marielle, and Jan also works as a PhD scientist at Kimera. When I went down to visit Miami, I was thinking I was going to move there. They were kind of to show me the lab, and in the lab they have these wonderful machines. Of course, everyone and their sister has the nanocyte machine. That's the one next to my head, NS300, and that'll count the particles. You don't know if you have meconium fetal skin cells, junk, debris. You just have particles. So they were really excited. I remember Duncan shared his picture with me. This is the ONI Nano Imager, which is an expensive machine, I think made in Germany or something. Anyway, it's you can get actually an image after you radio tag them with these, luminescent antibody markers, I presume. 63 and 81 can be shown, demonstrated. Yeah, these are exosomes. Not only are they the right size and particle, but they got our standard tetraspanin molecule markers there. So that's exciting. You know you got the real deal, at least by what's in the canonical papers. Duncan is a PhD in immunology, and he has many years of culturing cells, going back at least, I think, to 2013. So he's no amateur winemaker, if we continue that analogy. It's been an FDA-inspected facility. They are currently in submission for 3 INDs with the Food and Drug Administration, and they employ 40 people. 5 of them have PhDs, and 5 have experience with getting products through the FDA. So we'll go to Marielle's slides, and she was so kind. Thank you so much for sending this to me. I asked her to present something, and she really exceeded expectations. So we'll go through some of her slides now and try and understand. The goal here was, of course, to standardize how strong the exosomes are, how pure they are, what's their identity, and what's their potency. And to do that, we need to get a little bit in the weeds with the science. So the first 3 things they did, well, the first 2 were protein concentration. One method was BCA, which I don't think correlates as well, as we'll see. And then they have adsorbents at A280. So I think this is a combination of the HPLC technique, and they look at the 280 nanometer length, which correlates highly with protein concentration or protein. So number 2 was the BCA technique. Number 3, particle concentration by NS300. That's everyone does that. You buy any competitor exosome, you're going to get that graph, the scatter plot, and you're going to know quantitatively how many particles you have, what the mean size is, what the mode is. RNA concentration by absorbance at 260 is also measured. So we see here on the upper left this colorful graph. We see that in the area where the light is 240 to 280 nanometers, there's a divergence based on the concentration of protein and nucleotides. Anyway, this is a little above my pay grade, but there are formulas, there's ratios. People have been able to infer, if you have a mixture of 100% protein or 100% RNA and you vary them, then there's different ratios that come out just because of the different adsorption of light in this area. So like, for example, you see here in the middle graph, you see something running through an HPLC tube at just before 10 minutes. There's a peak. That is the peak of protein. Yeah. Okay. So a little bit heady stuff. Something that we need to get into the weeds a little bit with is this Purity, number 5, product purity by size exclusion. That's the part I really didn't understand. We were just on the phone talking about it. Identity is pretty much easier. That's the CD8163 Western blotting. Identity for RNA agarose, that's that screenshot. And then finally, they did an active bioassay, which TNF alpha is a marker of inflammation. So they measured the modulation of inflammation by different competitors' products. And dun, 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 drum roll, please. We got the results. So I had to personally redact all the competitors because this is not a hit piece. I'm not trying to shill for Kimera. I don't know if this data is reproducible. But anyway, here we have it. So we have Kimera's 2 products. Now keep in mind the Kimera Pro product is triple strength. Typically, people report billions of particles per mil, so they intentionally have triple strengthed it. Nonetheless, you have their exosome product, you have the amnio product. We have company 1 has 2 products, which are redacted. Company B, I'm sorry, A and B has 1 product. Company C has 2 products. Company D has a lyophilized, which means freeze-dried product. And these will be the main contestants on our game show. So we look here and we see total protein concentration. All right, going down the columns on the right, again, we see this BCA method. It correlates well for the Kimera brand, but it really doesn't correlate well with the other 2 measures of protein in other brands. So maybe we exclude that, maybe we don't, I don't know. In the middle column is SEC, that's size exclusion, HPLC. And then on the left is that standard protein concentration by A280. So, you look across, they all have protein in them. The one, company C, seems to have you have 3 decimal points, so it's a little confusing to read, but basically we're looking at, for company C's product, 35 micrograms per mil, and that's consistent across all 3 measures, and it's quite low. Whereas the Kimera one is nearly 1,000, 952, 887, 887. And keep in mind, you don't want a lot of protein necessarily. That's, as we'll see from the calculation, protein dilutes the potency according to the Ross unit, which we are debuting here. Okay, particle size. This is commonly available data using the nanosite. So what we see here, very interesting. The mean, the average particle diameter is 115 nanometers, which is a little on the big side. The mode, which is the most frequently occurring number, is 121, which is a nice, juicy, healthy exosome. And the particle concentrations per mil is shown there in log form. I'm sorry, exponential number, or whatever they call that. So we look at products from company A. We have gigantic particles. They're twice the size. And I remember I had at least a 20-minute discussion with the company founder on that. And he argued, bigger is better. And of course, if you go by whatever is 4,3 pi r cubed, the bigger is much better. But is it really, if you're using a bead agglutination and they're grabbing apoptotic bodies or microvesicles that are not exosomes, then yeah, maybe it's not better. Maybe it's better to just go with what the scientific literature says is an exosome, which is a prefab, again, on the order of the coronavirus, 120 nanometers, made by the endoplasmic reticulum, packaged in the Golgi apparatus as a bundle. All these big ones maybe not be better ones. And certainly you have to worry about bead agglutination. Company B is probably the closest competitor to Kimera in terms of what they're producing as a product. The mean is 145. Company C has 2 these products. Again, on paper, as far as the nanosite goes, pretty good. They're a little big, which big is not necessarily better in this case. But anyway, we're pretty comparable here, unless and until, this is another redacted slide. We see company A has 2 products, and I would argue that the amnio-derived products have a lot of particles. They have a lot more particles. So we see company A, product 2 is orange. That, again, is an order of magnitude more particles. Even Kimera's pink product, amnio 2X, a lot of particles. Well, when you do an amniotic fluid exosome product, you're probably getting a lot of meconium fragments, which is baby poop, fetal skin cells, other debris. It's not the same kind of thing as you have whatever's secreted by the cells in the media. It's not as pure. So, and then as one of the slides here will show, there might be more cells in amniotic products as well. We look at the exosomal RNA concentration. These are different methodologies. Again, we have micrograms per mil. Kimera seems to be ahead with their triple strength product, although it's a pretty close race. Now they try and look at percent purity. The ratio of RNA to protein is a conversion factor. So you see 89% purity, 35% purity, 60% purity. So multiplying the concentration times the purity, they get a corrected RNA concentration, which is logical, scientific, and makes sense. So these are some of the numbers, and I've tried to go through them and validate them, largely validated on the last slide, which is the final standings. When we look at RNA identification, very, very concerning that in the competitors' products that are redacted there on the left, we don't see the RNA agarose gel. It's not showing all that RNA for some reason. On the right gel, we see various lots of the Kimera products that are quality tested, and they are showing the appropriate bands for RNA. Here are some of the actual photomicrographs of the radio glowing tagged CD81. And then Marielle put a comment here, the bands observed in amnio and the other sample apparently coming from cells and not from exosomes. Similarly, when we look for CD63, we see again the same comment added by her, the bands apparently coming from cells and not from exosomes. So the take home, I think one of them is maybe don't use or think about the repercussions of using an amniotic-based product, because you might not be getting pure exosomes. You might be getting a lot of other stuff, including cells. And finally, they had that active bio test. The green is increased inflammation, I suppose, and one of the company C product 1, showed an increased inflammation, which is of concern. Other products seem to show a diminution at a higher concentration, so dose dependently of tumor necrosis factor alpha. So the big reveal, the unveil is, well, if we don't want to talk about billions of particles, if we're tired of answering questions of why don't you just get more for less, then what we have to do is find a way to quantify. And so what we've talked about this last, 40 minutes or 30 minutes is what's in them. Okay, so it's not that easy a question. First of all, do you know how to separate cells? Maybe you do, maybe you don't. Maybe you've never tried because you're still running on the same old cells. Number 2, do you know how to husband them, like keep them from differentiating, know when they're bad, know when to replace them? Number 3, do you know how to test them? You can't just send it to your 24-hour photo and test them. You got to have standard kits, okay, or develop your own protocols to open up the exosomes, crack open the toys and see what's in them and analyze them. So it's a huge scientific experiment and process to come up with this formula. So basically what they have is a RF on the right, Ross factor, and then the other stuff is just basic math. It just makes sense. You have, concentration. The more you have, the more units you have, right? So V is volume. Okay, we're okay with that. We don't know what RF is, but V is value or volume. Now here, actually total protein is bad for the Ross unit. So if you have a lot of protein, that will inversely correlate with your Ross unit potency. So that's the only one in the denominator. In this numerator, the more RNA you have and the more exosomes you have, the better. So that's all pretty logical, I think. So basically they're saying RNA times exosomes divided by volume is basically it. And then you multiply by your amount of volume. So I will probably not, well, I might if I'm forced to talk about Ross units, but we've been talking about billions. So that's kind of like the way it goes. But the frustration is, are you really getting a billion functional exosomes? Maybe not. But anyway, for those that are used to the old regime, there's the purple product, which is regular strength, and as I mentioned, the blue product is triple strength, so triple concentrate. So roughly speaking, 10 Ross units is 1 billion. If you're talking about the Kimera product, 50 Ross units is the 5 billion. Roughly speaking, if you get the super concentrated 1 mil, that's 30 Ross units, is what we used to call 3 billion. And for the gold standard, we have 5 cc's of the triple strength, that's 15 billion, 150 Ross units. I hope I did that some justice. And the final slide, the pièce de résistance, is the redacted company comparison. Target value on the right, Ross unit value. Again, now you should understand that 147 is pretty close to 150. 150, 10 Ross units per billion is 15 billion. When you're talking about amnio products, like you just throw that out the window, I think, but that's a different topic. So their 1 cc regular concentrate version is coming in at just under 10 billion, 1 billion, just 8.5 Ross units, which is about a billion pure exosomes suspended in saline. When you look at their products, competitors, Company A has 2 products, and the Ross unit value is quite low at 0.8 and 1. Why is that? Not really sure. Company B has probably the most similar product. They're coming at 2 Ross units, so you'd have to do the math if you think that that's worth it in terms of their, I don't know their costs, but they do have what appears to be a somewhat functioning product. Company C, as I just talked to Marielle, and we'll get her on the phone, it comes in at 0 concentration. Of course, you multiply anything by 0, you get 0. So they really get a raspberry there at the Ross unit value. Is that a fair methodology? Well, according to the adsorbents, there were no exosomes by their size exclusion HPLC. So I'll leave that to you and to Marielle. And finally, the lyophilized freeze-dried product, different sort of thing altogether, coming in at a Ross unit of 0. All right, I've talked enough. So let's talk about what's the conclusion. What is all this, Dr. Park? I'm confused. I don't understand. I kind of get it. In Latin, they say caveat emptor, which means buyer beware. And if I'm going to buy something, I got to beware of whether it's going to hurt my patients, help my patients, be too expensive. Are there other competitors? But then again, I've had friends switch from Kimera to other brands, and they say it's good at first, now it's not good. Who the heck knows? It's all a very heuristic thing, but we have to back it up and think about how difficult it is actually to do cell husbandry. But assuming these data are correct and the methodologies were fair, like they didn't treat the competitor samples differently, they did all the same protocols, then the Ross unit characterization appears to denigrate the quality of the competition. It's at least 20 times stronger. So that's just kind of mind-blowing. They're not sumo wrestlers. One's a sumo wrestler, and one is a, Tiddlywinks player or something. Anyway, if the data are correct, and there is an explanation, I would probably say that they don't know what they're doing in terms of taking care of the cells and measuring the quality. It's to be expected that cells will decline, as shown above me. Standard chart, yeah, you can get a lot of cells from 1 placenta, double, double, double. There's the logarithmic growth. But towards the end, funky stuff starts going on. It's like there's too many cells, they're overcrowding, there's contact inhibition, they're aging out. They usually don't lose their telomeres because they have telomerase, but the epigenetic expression, gene silencing, they're acting old, they're bad. You got to throw them out. They're like these chickens above. You don't want to eat eggs from that chicken. So I just think they don't have enough PhDs there. They don't have 40 employees and 5 PhDs. They just don't know what they're doing. And when you don't know what you're doing, you're kind of scary because you're making mistakes. You don't even know if they're mistakes. Anyway, I think that's probably the problem. So in a cell culture, you anticipate that the cell lines, although immortal, will become zombies, again, by epigenetic and genetic drifting, gene silencing, senescence, and contact inhibition. So I believe that the companies without the expertise, they have this large set of unknown and unknowns. They're just trying to make money. God bless. And why would I risk trying a product that may not work, may not be a better value, and might even hurt my patients? So that's not where I'm at the current time. I'm not morally opposed. Again, I didn't get paid to shill for Kimera, but I just not within my risk benefit analysis right now to do that. But I'm open to hearing about it. So anyway, I think that they're doing a good job assessing the quality. I think they're monitoring it. They do internal reviews. Now they've done a competitor reviews. The results, if they're to believed, are quite shocking. And so I think that taking these adult, because from what placenta, they are adult, they're not embryonic, the ability to grow these periodically, new batches, is not trivial, and it's very hard. And the ability to separate the exosomes from that solvent and then validate the contents. These are all incredibly difficult things, requiring scientific skill sets, prefab kits, rigorous methodology. It's not a joke. So when I look at the people running, and I've talked to the scientists and owners of a lot of these companies, and I start to get into the technical stuff, not because I really know or am that interested, but I just want to see what their level of comfort is discussing the science. And it can be quite shocking, the kind of stuff they try and get away with, thinking that you don't know what they're talking about. So anyway, I hope everyone's enjoyed that. I'm going to turn it over now, close the presentation. Marielle, Duncan, if you need me to open the slides, let me know. Hopefully everyone kind of has a visual memory of that.
Hi, thanks, Ed. I was just saying that, we're going to ruin this unit concept if we correlate units to billions of particles according to the ExoGlow product, because that, again, is a setting on your nanosite. So while we say a billion, we can easily change the settings on the nanosite, and it's 10 billion or 20 billion. I mean, you can make that nanosite read whatever you want, and our competitors make it read as many as they can because it sounds good, right? So I would just like to say that we have X units in the regular and X units in the pro, and just leave it at that. And we sort of have to re-educate people. I know it's not comfortable, but I don't want to discuss it as directly correlated to billions of exosomes anymore.
Absolutely, yeah. So if we have to move to the metric system, we will, or whatever. Marielle, did I destroy your data?
No, I just wanted to clarify if anybody has some questions. You were pointing about why the ROSE unit for some of our competitors were 0 or very low. And the main here is we have to, in the way that we create the ROSE unit and we evaluate our product, is one method is not better than other. Everything is creating a puzzle to finally define the quality of your product, taking in consideration safety, identity, purity, and potency. For example, we do the concentration by 880 BCA and HPLC. If you remember the slide, the Kimera product, all the three methods are comparable because our product has the protein from the formulation and the exosome products. Those three methods are not comparable is because they have additional components, or either as part of their formulation or are impurity from the process that they absorb at 880 and they are not proteins. For example, it happened with the amnio product. Amnio has a vitamin 12 peak in the HPLC that absorbs at 880, but it's not a protein. Then the BCA value is lower than the 880 value because the product has other components other than proteins. And then that's why we put the three methods together as a comparison, because it will show you also how pure is your product in comparison with the others. The second one is also the concentration of exosomes. We always evaluate that by HPLC, we are able to identify the peak of exosomes that we have been defining in all of our product. It has a relative retention time to an HSA peak. Then whenever you find that peak in the HPLC, then initially we assign it as exosomes. Size exclusion is not a specific method. Then anything that has that molecular weight will come at that peak. That's why it's not HPLC by itself. We compare that with CD81 present and also RNA present. Because we even noticed in one of our competitors, for example, they have a very little peaks that absorbs in that area, that if you put it together, it might give you a higher concentration. But when we did the RNA extraction, it didn't have anything. And in the gel, it didn't show up anything. And in the CD81, we were able to see that there is a non-specific interaction. Then therefore, it's not like those particles or those peaks, they were really legitimate exosome peaks, but we consider, that's why we put all the factors together to consider the total protein concentration, like a formulation that the product has, but we confirm that are exosome because of the RNA content that is present. And also that peak, that should be a single peak. It should not be an agglomeration of peak, because also if you have impurities that they aggregate, and you will have this disturbance peak, that they are not really a beautiful and symmetric peak as we see with the Soglopro. Then all of those conditions together are the ones who make the puzzles to come to place and be able to define that the product that you are generating is an exosome product, and also the quality of it. If it's a pure exosomes, which is the percentage of purity that you have. And also after we produce, we also evaluate that our product has the parental quality, that we evaluate with particulate metrics that is not present there, and then sterility. Then it gives us a confident scenario that our product is safe for patient use. And I want to do another remark about what you mentioned in regards to the several placenta. Every time that you run a different placenta, is not only the safety test that is recommended to be used. We need to qualify one placenta versus the other. Then at Kimera, we evaluate the presence of all the surface markers, CD44, CD73, CD105, CD190. Some of them, they have to be negative. Some others, they have to be positive. Also, it's important to be able to, you have to define that you have the 3 differentiation path to make sure that you are generating an MSC cell line. Then, and also the morphology that is evaluated. Then the more, I mean, it's really hardly to believe that when you have 20 or 40 placenta evaluated in a year, you will have time to do all the proper characterization to make sure that the process, that each placenta are comparable. Therefore, the exosome probes that you are going to generate, they will be comparable. That's why we try to have only one master cell bank. In this way, we know that lot to lot has really high percentage of being comparable one to another. Then the protocols that you doctors create to generate to treat any indication, you know that the product that you are acquiring will work in the same way.
Yeah, I think that's something.
Patient.
Yes, thank you. Beautifully said. I appreciate that. As we discussed briefly when I visited you and Jan, the intellectual property of owning a cell line and its products is murky at best. But I think what you're trying to clearly make out to us is that it's not that easy to get donors from 40 placentas and make sure that they're consistently the same quality.
Well, not only that, but what the FDA requires us to do per placenta is about $150,000 worth of work. That's outside of the internal characterization work we do. So the concept that you can just use 40 placentas per year is something that people do, but it's absolutely not legitimate. And that's not what the FDA is wanting or expecting you to do for approval.
That's correct. I wanted to turn it back, and thank you, Marielle, for that explanation. But, I got a question from someone who couldn't attend named Scott, and he asks, how long can you use this 1 placenta? When are you going to run out of cells? Can you answer that?
Well, it depends on the master cell bank that we generate. Whenever we treat the placenta, we try to generate as many master cell bank vials possible. Then we create from that master cell bank a working cell bank. Then, for example, we are trying to see at least if we can have at least 5 years worth of product in any master cell bank before we go for the immortalization. In this way, we know that we have time to qualify a new placenta, to create a new comparable master cell bank, and then that will generate comparable exosomes.
I see.
it's not a number set of stone. Each company has their own path based on sales, based on expected indications and so on, but that's our plan. We are aiming to have a master cell bank that will last as long as possible.
Yeah. So, turning back to the presentation, there was a lot of stuff that was quite frankly above my head. But one concept I see a lot in textbooks and articles is that when you are growing cells, there's a logarithmic growth, and then there's a decline. Have you been able over the years to observe a decline in a certain lot's quality towards the end of keeping it?
Well, we try not to extend the cells for a long time, and that's how we evaluate the stability of the cell line. Then we always treat our product in a certain range to make sure that we are always at the plateau and we are not in the decline area. But every lot that we produce, we do the same quality control to ensure that all the qualities attributes are met. If for any reason any of them fail, then that lot is never released.
Yeah.
Then it will never be going to the market.
Right, but I presume that because of filtration or whatever method you're using, that some of these, like for example, your competitors has 250 nanometer exosomes, right? I presume this is a post-isolation quality control, so you're not going to see your large abnormal exosomes or apoptotic bodies. So the question is, does the actual yield start to decline towards the end of a life cycle of a lot? Do you get fewer eggs from those chickens, so to speak?
Well, that's why you establish the expiration date on your product. I have seen many competitors.
No, that's not what he means. What he means is if we grow, we only use the cells for a month. Okay, that's what we've done and we've always done.
Oh, okay.
If we use those same cells for 3 months, then you might start to see a drop off, right? Right now, we're actually taking the cells out, not for production use. We're just doing it for fun. We're seeing how long the cells go out and trying to compare it to earlier time points. But in order to avoid what you're saying, we have just always given up in a month. Throw it out, start over again. And that works fine, and we've created a reproducible product that way. I'm sure that if we were cheap, we could extend that a longer period of time, but we're not.
So, Duncan, assuming that the math and the methodology is legit, what's your explanation for why the competitors' products don't appear to measure up in terms of potency?
Well, everybody
because they don't know that. I mean, they think that they're doing a good job, right? Because they don't have all these characterization methods. So I remember how scary it was. The reason that mine has always worked is because I did it from a number of cells, and I just did it the same way. I wasn't tasked with competing with myself. So I didn't just follow a recipe and see it working. I had a product that I saw working, and then I characterized it. When they characterize it according to the current methods that they have, they think that they have exosomes, but they don't, as I just found out. So the difference is that mine were always therapeutic, and I kind of worked out that way. I didn't have to work backward to compete with Kimera. Right? Does that make sense?
Yeah, it does. But, I've heard some concerning things, people getting very inflammatory reactions, a lot of variability between potency of certain batches. Do you think that other competitors are not actually changing their batches every month?
Well, I know one competitor for sure was making it with one provider, and it was really just a dilute bone marrow from fresh bone marrow or Wharton's jelly. And then they switched to a new producer to try to produce it the right way. And then that producer has had trouble learning how to do it. So you're getting the different product every single time, and thus you're getting these different outcomes.
That's funny because most people would just naively think, well, I roll up to Shell or I roll up to Exxon, I'm going to get gas, and there's a certain internal quality control. What's the difference? But I guess the devil is in the details. Were there any glaring omissions or errors in the presentation that you want to point out?
There was one more thing about that I found interesting is that we do different types of in vitro assays to tell whether or not a product is anti-inflammatory.
Right.
And the TNF alpha assay is fine, and it shows that, like I've said in my talks, that amniotic fluid products do bind up TNF alpha because they have TNF receptor and they have IL-1 receptor. So you might be confused if you look at an exosome versus an amnio product. You'll say, well, they're both anti-inflammatory, but they're both anti-inflammatory in very different ways. Like I've always said, one is temporarily anti-inflammatory and the other one's permanently anti-inflammatory. We're moving to a new assay using IRAC1, which I'm not sure you showed, which is a downstream protein in the inflammation cascade intracellularly. And if you turn off the inflammation intracellularly, which we've demonstrated that our exosome does, then you know you're really turning off a system-wide inflammation rather than just sponging up inflammatory cytokines in the milieu.
Yeah. Well, I think it's very important. Marielle obviously has a very rigorous scientific mind, and I think she was trying to bring up a point the other day the other moment about vitamin B12 peak correlating with A280. This is way over my head, but I guess what the point is, you have to have integrity. You have to have honesty. Look at the data, and no matter where it falls, you have to do a methodology that can validate both your products and your competitors' products. And I guess the take-home lesson would be that, according to the standard methodologies and the measurements you came up with, there really is no competition out there right now that is similar. Is that a fair assessment, Marielle?
Well, in our case, we were including this ROSE unit even before we evaluate the competitors, because we wanted to have a method and a strong method in-house that was not just a nanosite, because we know that particles doesn't have any real meaning in terms of the quality of the potency of the product. That it was the intention of the ROSE unit to generate a unit that was robust enough to generate the strengths to characterize an exosome product, no matter it was Kimera or any other product, that will contain the main factors that are evaluating because the exosome is not like a monoclonal antibody. That is only one structure, and that's why we just do concentration. The exosomes has the RNA component that is really implying a lot in the biological activity, but at the same time, you have the concentration of exosomes that initially was particles, but the particles are, if you have impurities, they are still particles, then it is not really reliable. And that's how Dr. Ross came out with a ROSE unit. And in Kimera, we were trying to see which were the factors that we should put there to make sure that we can compare lot to lot at Kimera to make sure that we are always producing the same reliable and comparable product lot to lot. And then we evaluate how these units will translate with the competitors. We were not even knowing how the data was coming up. We were putting all the samples together, and we were really surprised that they were not at all comparable.
That's the take home. I was surprised too. I thought it would be like similar, but I don't think it's similar at all. Anyway, so we have a couple of questions from the audience, if you don't mind. One is this available in the UK, for that matter? Duncan, are you available in any other country besides the US?
Well, the UK is probably the next one that we can get to, especially since they've now broken off from the EU. So
the answer, UK probably, but currently no other country. You do have an IND, which means you can ship across state lines. So I guess US is the only game in town right now, correct?
For the next couple months.
Okay. And I have a question here from Christine. She asks, as the cells are expanded, do they lose their stemness?
This is again, as Dr. Ross mentioned before, we have a time frame for evaluation, and then we do the expansion always at the same passage number, and the cell culture of production, the same passages number. Then we don't allow the cells to decline because we are working always in a very short passage. And then we also monitor our cell culture, not only visualizing the cells, but also we evaluate the metabolites. Then we see how the lactate is moving, how the glutamine, the glutamine, the glucose, and all of those levels also associated to the cell culture. In this way, we can monitor also if the cell culture, for any reason, is moving out.
Right, that's right. So if the cells early on are using up X amount of glucose because they're healthy, and we leave them in there past the month to 2 months to 3 months, when do they start using less? glucose? When do they start, not producing the metabolites that they should produce? We're doing that just to characterize it, but
Right.
that's something that can be done. Right?
Yeah. So, like you would expect, like has been described in the literature, you would predict, Christine, that maybe not the stemness, but the quality of the behavior and exosome product by inference would decline over time. But they never let it go to that point. It's kind of like drinking milk after 3 weeks. You're only going to keep it for a week and it'll still be good. You don't actually push the limits of how long that lot will be producing your product, I guess.
And remember, we always, before the lot is released, we always evaluate all these quality attributes. Then even a lot that is the fresh batch, it doesn't, for any reason, it doesn't meet the criteria that we have in-house for release it, it never leaves the quarantine status. It could be the first lot of the batch, could be the last one, could be any lot. It goes through the same quality control.
And have you actually rejected lots in the past?
Yes, we had few lots that we have released that we knew because of which was the reason why, and they are still in quarantine in-house.
Right.
Never that is not meeting the acceptance criteria that we have in our specifications, it's going to our clients.
So you have declined to release.
Well, there's lots of reasons that you reject lots. I mean, there could be too many particles that come off of the vials, and then we have to reject it. There's all those types of things. Right?
Oh yeah, I'm not saying it's a bad thing. I'm actually saying it's a good thing.
No, but the inverse is. I think what the present person was asking was if you replicate cells too many times, do they go bad? And certainly they do. If you replicate cells in a bad medium, they'll go bad in 3 replications. If you use a better medium, they'll be good until 10 replicates. But we try to stay at 4. We make our master cell bank at 2 replicates, and then we make our working cell bank at 4 replicates, and then we don't go past that. Then we throw it out after 30 days. And what matters here is if you're getting the same output of, say, TGF beta 3 from passage 4 as you were from passage 2, then it doesn't matter whether it was passage 4 or passage 2. Where you're getting the end result, which is the cytokines that cause growth. And that's what we have to quantify.
So generally speaking, you're still very, very early in the infancy of what would be a viable cell line. And that's where you're harvesting them. Yeah. So do you keep them
So what people are afraid of, and what I think that my competitors may be doing, is letting those cells grow out for long, long periods of time. And that's why they're not producing exosomes anymore, because they know that they have cells, and so why aren't they getting exosomes? That's one reason. Right.
Yeah. So they have like menopausal stem cells or something.
Exactly.
It's the same criteria that it should be used for any cell culture. It's not only for exosomes, for monoclonal antibodies, for any biological product. You always establish your acceptance criteria for harvesting the culture. Then even if it's a suspension cell, you have a bioreactor, then you go usually you establish criteria in viability and metabolites, and whatever is failing first, then you harvest your culture. And that's why we also put the metabolite, because, for example, maybe we are still within the frame that we established for our cell culture, but if there is any spike in any of the metabolite that is moving out of the trend, then that culture have to be harvested. It's not like you sit on a stone and you don't monitor your culture. You always have to monitor to make sure, because biologics is not 1 + 1 = 2. It could be 3, it could be -1. Then you always have to be very observant and put all the in-process control evaluation in order for you to be able to detect if any abnormality is happening and to be able to take the measures accordingly.
Excellent. All right. If there's no other questions or comments, any other questions, Duncan, Marielle, audience?
No, thanks for your time.
Yeah, no, I appreciate it.
Thank you very much.
Yeah, it was a long one, a lot of science, but I hope I didn't misrepresent what you had found too badly. I think it's...
No, you did a great job.
Okay. So no big errors there. Okay. All right. Good. All right. Well, everybody, thanks a lot. I hope this was somewhat clear. I know there was a lot of science, but basically my gestalt of it is that I don't think a lot of these other competitors have the ability to even know if they're making a viable product or not. So Remember, it's just not all about the money. Okay, thanks everybody. Dr. Ed Park from Recharge Biomedical. I want to thank Duncan Ross from Kimera and Marielle Gonzalez as well. See you next time. Thank you.
Thank you.
Bye-bye.